Thank you. First, I want to thank the organizers for inviting me here to talk today. Hopefully, I can offer a perspective from a basic scientist that's really been working in the HIV field from a bench to bedside and bedside back to the bench kind of approach. So for today's talk, I really wanted to focus on kind of three parts of the talk. The first part was really informing the group about the field of geroscience and how the aging field has really reconsidered some of the mechanisms for some of the potentially targetable aspects of aging that they're interested in studying, and the framework for how the aging field in general is thinking about studying the aging process. I'll overview a little bit about my expertise in terms of epigenetics. This is really a field that started in developmental biology, but now as we start to think about how the environment and all these myriad of factors that we live in are impacting our genes and how they're regulated, this is really that field in terms of trying to understand basic mechanisms. I'll update the group. We learned a little bit yesterday from Kaylin's presentation about epigenetic clocks, but you're going to start to see this, I think, even more than we already have in the popular press. And in terms of as we start to think about clinical trials and how the field is wanting to measure if we're having an impact in terms of some of the mechanisms we think that may be leading to accelerated aging or attenuating aging. In part two, I'll tell you about this interesting project where we really reconsidered these epigenetic clocks and thought a little bit more about what we know about HIV and working with some of the projects in terms of antiretroviral therapy and thinking about some of the epigenetic clocks that, for the most part, were built with machine learning methods, but the aging field was kind of stumped in terms of what they're actually measuring. They knew it was DNA methylation, but none of the genes that they were able to identify had anything to do with the aging process and really weren't targetable. So we went back and reconsidered in terms of our genome endogenous retroviruses and thought about potentially building a retro clock on that aspect. So I'll tell you a little bit about that project. And in the last part, I'll just briefly summarize the challenges in terms of identifying gerotherapeutic interventions. So geroscience as a field is really focused on studying genetic, molecular, and cellular mechanisms that make aging a major risk factor and driver of common chronic conditions and diseases of older people. And really, the field of geroscience has really gone back to the basics in terms of thinking about cellular processes that would be leading to a myriad of complicated comorbidities rather than focus on a single comorbidity as a focus group. So for example, if you're interested in Alzheimer's disease research, rather than just studying the pathology of Alzheimer's disease, it reconsiders potentially what cellular factors or epigenetic factors may be contributing to that, because typically in that particular disease context, Alzheimer's would not just present as a single disease. There'd be a myriad of factors that would need to be considered. In terms of the hypothesis for geroscience, and really this is what the field is working towards trying to address, it states that slowing the aging process potentially can improve health and delay the onset of chronic diseases. So the aging field, really, I think in the lay press, we're seeing a lot of this rejuvenation or stopping aging or even mortality concept. But the reality is, I think the majority of the field is really focused on improving healthy aging. So not necessarily completely stopping the process, but making sure that that process, is a lot better for everyone. In terms of the connection with HIV, I think there's a very unique opportunity here to mesh these two fields together, and this is really, what I'm excited about building my career in terms of trying to identify those mechanisms that lead to age-related diseases. In terms of the left side here, this figure is showing from the geroscience perspective these hallmarks of aging that are being studied. One component of this are epigenetic alterations. We have other things that we've read a lot about in terms of telomere length. You have mitochondrial dysfunction, nutrient sensing. And if we overlay this in terms of what we know about those that are studying people living with HIV that are aging, a lot of these factors can be identified and studied and kind of frameworked in this geroscience hypothesis in terms of the mechanisms of aging. In terms of epigenetics, very simply, this is the study of how our genes can be turned on or off. And when we think about it, every cell in our body essentially has the same DNA code. So the same particular pattern of genetic material is laid out in every single cell. But as we know, these cells change function and respond to the environment and interact differently in terms of a myriad of factors. So very similar to a music score, it would be as if a conductor or the environment throughout time or throughout someone's life is essentially in pencil or pen writing down particular marks. And these are the marks that we're able to measure in the lab and identify and associate with a myriad of different disease context, but also phenotypes that may be protective in terms of the aging process as well. In terms of these marks, or if we think about these pencil notations that would be laid down on a music score, the marker that I've spent a lot of my career studying has been DNA methylation, which we've utilized mainly because it's a covalent modification to the DNA code, so it's quite easy to assay in the lab and stable. The other fascinating part about DNA methylation is there's 23 million different positions in our genome that this mark can be assayed at, but only 1% of it is really the dynamic portion. So a large part of our genetic material is established for a particular cell in a particular context, and it's this very small percent that is where the activity and action is that we're able to identify. And in terms of this mark, this is really what was utilized to develop these epigenetic clocks and stumble upon kind of this phenomenon of having a potential biomarker for aging. In terms of the context of applying these epigenetic clocks to people living with HIV, I'll briefly summarize what we know. The first thing is a subset of people living with HIV seem to be accelerated or advanced in terms of these epigenetic clock estimates. I always like to emphasize in that it's a subset of people because if you actually look at the data, we do find there is a large proportion of people that are doing well on these estimations. A lot of these publications and reports we're really finding mean differences. These are statistical differences, but if we look at individuals over time, it's actually quite fascinating. So if we were to take my epigenetic age and look at it over time, for the most part, there's a lot of stability in these estimations, so it's quite intriguing in terms of identifying someone's actual aging biology is relatively stable over time. And in terms of these cross-sectional differences, we also find that people living with HIV reduce their epigenetic age on antiretroviral therapy. So we do see, a slight reduction in terms of a group difference as well. However, one limitation of these prior studies is their lack of cell type specificity. And our lab really has focused with working with immunologists and understanding the biology of HIV as well in terms of focusing on particular cells in the body that could be studied. When we think in terms of what we know, CD4 T cells would be a quite important cell type to look at, as well as potentially these immune cells that are inflammatory, which would be potentially a monocyte cell or cells that would produce inflammation that may be lingering. And the other aspect, which I had this unique opportunity, and I'll summarize briefly, is a project where we were able to ask how potentially early these epigenetic age estimates may be increased in a subset of people. I was quite lucky in my career to get involved in a project with my primary mentor that was a partnership internationally in Thailand with the US military and some investigators throughout the US where we're able to identify people living with HIV during acute infection, so the earliest potential time point. And in this project, they initiate antiretroviral treatment that same day. In this particular context, we were able to look at epigenetic age in two populations, so these monocyte cells and CD4 T cells, and ask just the simple question, how early are these impacts occurring? In terms of what we found, immediate initiation of ART first reduced epigenetic age in the CD4 T cell compartment. Here I'm showing you data comparing those individuals pre-antiretroviral treatment. These individuals potentially were captured within 2 weeks of acute infection. In terms of the monocyte cell population here, just briefly summarizing, what you see is despite antiretroviral treatment, there's this lingering signal of increased epigenetic age or this inflammatory signal that persists despite antiretroviral treatment at the earliest potential time point. Suggesting, I think as we start to think about these studies and looking at where we're measuring them, we really need to consider partitioning out potentially cell types that may be having particular aging signals more so than other cell types. Because if we were not able to do this at this resolution, we would capture a bulk signal, which may still be informative. However, it wouldn't provide us kind of where the cell type to potentially focus on in terms of reducing or ameliorating some of these changes. And just these findings at the early stage support some recent work as well that was published in the MAX and the early work that was published as well. And in this MAX study, they were able to capture participants in the MAX study before and after, so acquiring HIV infection in that particular context. So just suggesting it looks like these changes are occurring early. There does seem to be some amelioration in terms of the CD4 T cell compartment, However, I think we need to think about potential other things to reduce inflammation, which I know a lot of groups are working on as well. And then, as I said, these estimations in terms of epigenetic age were, at the most part, working with people that have been doing a lot of machine learning. And what they're doing is using this data set and big data and then identifying things that are reliably predictable of actual chronological age. However, when you ask the question, well, what can you do to modify these changes? There are drugs in the cancer field that impact DNA methylation as a process. However, these drugs are globally impacting methylation patterns. So the impact of that in terms of what it would do in terms of modifying epigenetic age or impacting the aging process has really not been clear. In terms of thinking about causal mechanisms, we got quite intrigued in terms of the aspect of endogenous retroviruses. So if you look at the human genome, the majority of it is not coding for proteins or things that the cell needs. The majority of it is actually ancient viruses and transposable elements that invaded the genome in the somatic cells and have actually been silenced by DNA methylation. And these other retroviruses are living in our genome and have basically been considered to be the dark matter of DNA. And there's been a lot of research recently to show that these endogenous retroviruses actually are playing a role in gene activity. So we went back and were able to work with a group that had access to a data set of DNA methylation in the population of 12,670 individuals, spanning an age from 12 to 100 years. We went into the actual data set and only focused on those retroelements in the genome and asked the question, could we use just that methylation pattern of retroelements to predict age, chronological age, in these participants? And then we validated it using a subset of around 2,000 samples. Here I'm showing you the data. It's quite remarkable. So we were able to find in our training data set and the test data set, just based on the methylation patterns of these particular retroelements in a blood sample, we were able to identify someone's chronological age, suggesting that there is some potentially targetable aspect of the genome that's being modified with the aging process. And we've started to now apply this in data sets where we're looking at potentially the impact of antiretroviral therapy. There was a lot of beautiful work and a lot of early work showing that reverse transcriptase inhibitors have a side effect or alternative activity on endogenous retroviruses in the genome, shutting off their activity. We're now starting to look at whether there's particular new classes of antiretrovirals which may be impacting these epigenetic age clocks and some other aspects. So in terms of thinking about the aspect of people living with HIV and working with those that are interested in new formulations of long-acting, there's some intrigue now in terms of the aging process in terms of looking at reverse transcriptase inhibitors. So this is an area that is open for investigation, and we're interested in looking at this. So just to briefly summarize, in the first section, I told you a little bit about the geroscience field and how they're starting to think about studying the aging process. I've told you a little bit about epigenetic clocks in people living with HIV. I still think there's a lot more work to be done there in terms of looking at this globally, looking at sex differences as well. But I think the field has started to now apply these epigenetic clock estimates. And then in the last part, I told you kind of about the aspect of learning from both fields in terms of the epigenetic field, the geroscience field, and the field of HIV and aging, and particularly thinking at aspects of aging processes that are related to retroviruses and thinking about how we may be able to study that as well. I'll just end with how the aging field is applying these epigenetic clocks. This is a slide from the aging field showing you a plot of all the methylation data sets on the right-hand side here. These could be dietary interventions, they could be pharmacological interventions, and they had epigenetic data or epigenetic clock data before and after the intervention. The first take-home I want to emphasize is if you notice, there's a lot of blank space. It's very difficult to modify epigenetic age or this biomarker. And this biomarker right now is being considered in a lot of these clinical trials as a primary outcome measure that they're going to be utilizing to see whether they can reduce this estimation in particular individuals that are participating in these trials. The second piece I wanted to emphasize are there are some things that are in green. For the most part, these are the things that we've heard about in this meeting that are really what are recommended: exercise and diet. So if you really want to have an impact on these epigenetic clocks, it really at this point boils down to the two best recommendations I can make are exercise and diet. Beyond that, I think in the field there's some interest in metformin, some other aspects. I think the jury's still out. We don't have really good data to suggest that or implicate those particular aspects. But just to show you kind of what is being done in the aging field, and I think there's an incredible opportunity to learn from people living with HIV that have participated in clinical trials that were initially, I think, designed for HIV cure or for addressing potential comorbidities. If we go back retrospectively and apply these epigenetic clocks, this is what we're very interested in doing, to see whether it's quote unquote a trial that maybe didn't have the outcome of interest, but we can see whether the aging process or some aspect of these clocks is being modified. And here I've just kind of summarized potentially a lot of the incredible work that's been completed that now can be repurposed or redesigned to address potentially some questions related to HIV and aging. So just to end today, I just wanted to emphasize some future directions and challenges. I think a major challenge is really translating kind of these epigenetic discoveries into therapies. I told you a little bit about these epigenetic clocks, and if we start to think about a retro clock, there is a potential there to develop a reverse transcriptase inhibitor approach potentially to modify some aspects there. But really, I think that's a major challenge for translating these into actual therapies. I think the second major point I want to make, I've been to a lot of geroscience or aging meetings. There really needs to be, I think, an inclusion of people living with HIV in terms of these geroscience clinical trials that are being planned. I think a large part of that funding portfolio right now is being shifted to people that are relatively healthy to try to reduce their aging to become immortal in some regard. But I feel like that's a conversation that needs to be had, and I think this is really working with the community and getting a voice into the room to be able to have those discussions. And then the last aspect, I want to really focus too on the positive aspect. I think we are quite interested in funding and developing and studying cohorts of people that are having a super aging phenomenon, or potentially even people living with HIV that are centenarians. I think these are the cohorts too that we're going to learn potentially a lot about in identifying these mechanisms of aging that are protective, or really what is going on that is a resilient factor. So I think having both sides of the equation really are going to be important. But I think these are also conversations too that need to happen with bench scientists, clinicians, and the community in terms of identifying those individuals that also would be potentially interesting for being able to be, I think, in the room to say, what particular factors, what aspects of the biology, a lot of things we could potentially learn for those type of studies. And then the last part as well, I've really, I think, had an incredible opportunity to do some work internationally. Here I'm just showing you a plot summarizing epigenetic data that's being generated or deposited. As you can imagine, it's a very similar story to what we know for genetic data. There's even more severe lack of diversity in epigenetic data. I didn't have time today to show those slides, but if you apply these clocks in Africa, in Latin America, there's a discrepancy in terms of what we're being able to see in terms of predictions. These clocks need to be recalibrated or included in these models. We need more data to be able to actually make, I think, good kind of studies that would be potentially, I think, informative, globally. So just to acknowledge, I've had an incredible opportunity, I think, to work with a lot of groups. And, I'd really want to thank both, the study participants for the study I showed in Thailand, my primary mentors, then NIMH for funding. And I'd be happy to take any questions after the talk. Thank you.